All right, so this will be our last recording for the cell, and I'm going to go through the early part very quickly because the cytoskeleton, you can read up on it. What I do want you to know, though, is there are three different types of cytoskeleton parts, I guess. Microtubules, microfilaments, and intermediate filaments. So we want to go through real quickly and talk about their functions. And I have them listed more than anything, but so you can go through those on your own. But some of the differences is what I want to spend a little bit of time on. So one is what they're made of. So microtubules are made out of a protein called tubulin. Microfilaments are made out of a protein called actin. And then the intermediate filaments are made up of lots of different types of proteins. One of those could be like keratin, like your hair and nails are made out of. So they all have kind of different looks to them and different functions. So let's quickly go through. Microtubules are hollow rods. So kind of the shape and motility are important on these. We're going to talk about chromosome division later, and then organelle movement. So the organelles in your cell float around. We always talked about floating around, but it turns out they actually are directed kind of by highways, partially using the microtubules. When we talk about microtubules, we also want the ideas of centrioles, cilia, and flagella involved in that. And they really have kind of a structure here. So what you're seeing over here is these triplets of microtubules. You can see they're in groups of three. And then a little bit different here would be the centriole actually will have two microtubules inside here, which they're not really kind of showing you, but that's okay. So what I want you to really kind of think about for these is it really has to do with movement. Animal cells only have centrioles. This is important in mitosis and meiosis. We'll talk about that later. And then cilia and flagella are also made up of microtubules and very similar on what they do for movement, but they act in different ways. So the cilia is seen down here on the bottom in a paramecium. Tiny little hair-like projections, and what they do is they act like kind of like an oar in a canoe or a raft where you're going back and forth, back and forth, back and forth. Whereas flagella tend to be a lot longer. There tends to be fewer of them, and they more or less act as a kind of a whip-like action. Both of them will have very similar structures internally, like to the centriole. All right, filaments, microfilaments are a little bit different. Instead of hollow, they are solid. Again, cell shape is a big thing. These are also used in muscle contractions, which we won't talk about much in this class. Also, cytoplasmic streaming that you will see in the lab, and cell motility. And then the last one is intermediate filaments. These are more for anchorage and structure than anything else. So it kind of gives you an idea of what's used. All right, we'll talk about the cell wall real quickly. Again, in plants, that's made out of peptidoglycan. In fungus, that would be made out of chitin. And they have multiple layers, lamella, which are just, you heard laminate, it's just kind of a thin layer or something on top of another. And I'm not overly concerned about that. What I want to talk about more is kind of the breakages between that you can see there, which we'll talk about a bit later. And that there you can see the opening is called the plasmodesmata or desma, plasmodesma for one, plasmodesmata for multiple. All right, so here's your extracellular matrix. And you can see here's a plasma membrane we really haven't talked about yet. And then we have all this cytoskeleton, this protein structure for support. All right, let's talk about how animals and plants kind of communicate. In animals, there's three different types of things that happen. There are the first two are really kind of holding them together. The tight junctions are places where two cells are together. And again, they're kind of locked. They prevent liquid and stuff having to come through. But they're just really two cells kind of sticking together called a tight junction. The desmosomes are similar. The difference here is that they are going to have little projections that go in to anchor the two. And then they have gap junctions. And what I want you to know and really key into this, and why I'm doing this, is the gap junction is basically kind of these openings between two animal cells that are allowing for information to be information being passed. So again, tight junction and desmosomes, both are just kind of holding the cells together. The difference is the desmosomes have intermediate filaments that are attaching it and holding it on together. And then the gap junction is where you're actually getting communication going across. And then in the plant cell, it's the plasmodesmata or plasmodesma, where you're breaking the cell wall that's allowing for information to be passed. And then there's a different way that information can be passed, but I'm not sure it's important for this class. We talk about that in bio too. But just understand that the plasmodesmata is where the ions and things can go across, so plant cells can communicate. And then the last concept, I just kind of threw it on the back end here, is to understand, we have prokaryotic cells and eukaryotic cells on the planet. So the question is, how did they form? Now, bacteria have been, the planet's about 4.6 billion years old. Prokaryotic cells showed up about 3.5 to 3.8. I think 3.8 is a new number they're using now, a billion years ago. And then eukaryotic cells showed up a lot later. So the question is, where the hell did they come from? And there's an idea behind it. If you think about what a cell looks like. And so I want to go over this. Autogeneous is not a term that you will see in the textbook, but I want you to know the term. It just means self-generated. So looking at some of these organelles, the idea is they are self-originated. Autogeneous means that you're getting this infolding of membranes. And so you're seeing things like the endoplasmic reticulum, maybe the nuclear envelope or membrane, the Golgi apparatus. Those things could have formed through this system. Endosymbiosis is a different theory, and endosymbiosis means inside, living inside. And so what they think has happened is one organism actually has moved into another, lived inside, and have lost some of its other functions. And so we think evidence of that is mitochondria and plastids or chloroplasts, that they at one time were their own individual beings, and then eventually they moved in to live with inside another organism and lost a lot of their functions and now are living together. The reason we have evidence of this is that mitochondria and chloroplasts have their own DNA separate from the DNA in the nucleus. So we think that basically eukaryotic cells came from prokaryotic cells, and these were two different theories, and they've combined them into one now. In other words, well, not combined them into one, but both theories are how all the different parts to a eukaryotic cell have come across. So the question still remains, if you haven't figured it out, where the hell did the first one come from? And hopefully we'll talk about that later when we talk about evolution. And that's recordings for the week.